Now we're going to shift a little bit away from the legal topic and go to a little bit more scientific perspective on AI. I'm very happy to welcome Dr. Isabell Twick to the stage today. She is Principal Data Scientist at the Janssen Pharmaceutical companies of Johnson & Johnson. She has a PhD in neuroscience and a bachelor in biology and mathematics. And today she's going to talk about data science and digital health in the drug development life cycle. The stage is yours.
Yeah, thank you very much. Hi everyone. I'm going to talk about data science and digital health in the drug development life cycle in Johnson & Johnson. First, I quickly want to introduce myself. I'm Isabell Twick. I have a PhD in neurogenetics, and in my last position, I worked as a lead machine learning data scientist in CareSyntax. CareSyntax is a Berlin-based company that aims to improve surgical safety, and I performed computer vision experiments and some risk prediction modeling based on electronic health record data. And now I'm a principal data science in Johnson & Johnson, and here I use machine learning to optimize clinical trial operations. So first, I thought I'd start a bit to give you some background of the factors that we think really help data science and digital health to be more prominent in biopharma and healthcare. One of those is that we're now able to measure biologic complexity in much more detail. So what I mean with that is we have a lot of data on the DNA level, on the transcript level, also on the protein level. We call this omics data, genomics, transcriptomics, and proteinomics. And this data allows us much better to really identify what's the cause of the disease, and also is a big drive towards more personalized medicine. Then, of course, another point is the COVID pandemic has really helped in data collection. We collect more data, we also collect higher quality data, so that right now we have trillions of gigabytes of diverse healthcare data annually. This can be claims data from insurances, electronic health record data collected in hospitals. And all this data helps us a lot to follow the patient in their disease journey, so we really know better how the disease develops over time. Then, of course, as you all know, there has been a lot of advancements in AI and machine learning, and real-world evidence methodologies, and also in computational power. And all this together has led to a lot of investments in data science. And this brings me to the challenges that we still have in the ecosystem. We still need a more robust, complete, and ethnically diverse longitudinal data. So what I mean by that is when we currently look at real-world evidence data, there's a lot of missingness. It's quite messy. We also see a lot of bias because some patient populations are not as represented than others. So we have to improve on that. The regulatory landscape is also, as you may imagine, still evolving. Johnson's putting in a lot of effort to work together with regulatories. One example there is here in Europe is the European health data space. So basically the push there is to collect data across borders and standardize the data collection in healthcare to basically have better quality data, also to use this data for analytics, for AI, to overall improve patient outcomes, increase the quality of care, and also to allow us building models on this data. Another challenge is the consistent deployment of models in healthcare systems. There's different reasons for that. Sometimes models are just trained on really limited data sets so that we can't easily apply them to other disease areas, to other hospitals. Sometimes frequently actually, it's also because the health systems don't have the IT infrastructure or the staff to really help us deploy and maintain those algorithms. And finally, one challenge is data privacy. As with recent privacy breaches in the healthcare system, we have to really make sure to protect patient privacy. So with that, I want to go to the main part of my talk. Here you see the drug development life cycle. For everyone who's not so aware of that, how drug development works, we basically start on the top right with the discovery phase. Here we really try to investigate the disease, try to identify the targets that our molecules could bind to, and then develop those molecules. Then we enter the CMC stage, which stands for chemistry, manufacturing, and controls. This is kind of a safety layer where we check that the materials are correct, that we check methods. And then after that, we'll start the clinical development phase. This is where we actually perform the clinical trials. And here we test the drugs in humans for efficacy and also for safety. And then after that, we are entering the launch and post-launch phase, where we keep monitoring our drugs and, for example, check if there's any more side effects in the real world. And for the purpose of this talk, I'm going to focus on discovery and clinical development phase, where I want to give you some examples how we use AI in digital health in each of the different aspects. So starting with target ID and validation, as I already mentioned, there's all this omics data now that really allows us to dive deeper into the disease and understand what's going wrong. So we make a lot of use of that to really detect what are potential targets of our drugs. Then in the next stage, in the molecular invention phase, we currently use generative AI to predict protein sequences from amino acid sequences. In the past, it has always been very difficult to kind of estimate how a protein would look like if it's diseased. So we have the diseased amino acid sequence, but we don't know what's the shape that the protein falls into. So generative AI is really helping us to predict how the protein structure looks like. And with that, we are able to identify like quite different obscure pockets that we didn't know about before, and that can potentially be quite good targets for our molecules. And depending on the protein structure, we can then also develop these molecules that fit those pockets. Then I'm moving on to clinical development phase. So here, first stage is precision medicine. What I mean by that is we really try to find the right patient for our drugs. And not only the right patients, we also try to find them at the right time. So sometimes it's very important that we detect diseases early. For this purpose, we have, for example, developed an algorithm for pulmonary arterial hypertension, which basically predicts the disease just based on echoes and ECGs. And to give you some more background on the disease, it's like a rare disease. It's very uncommon, and it's usually diagnosed 3 to 4 years too late. So with these algorithms, we are able to detect the disease 3 years earlier. And we do it with data that's also already available, meaning these ECGs and echoes, they're collected anyway in those patients, just the diagnosis is often missed. So we can basically use our algorithm, run it in the background, and detect the disease 3 years in advance, and then, of course, also give the treatment to the patients much earlier. Another example for precision medicine is in one of our vaccine trials. It's called XPEC. Here we developed a vaccine for an E. coli infection. And this infection is very severe. It often leads to sepsis and often also death. So here, there's some risk factors known, which is mainly elderly people, immunocompromised people. And we really wanted to understand if there's additional risk factors that our patients have that could indicate whether they will get the infection. So we used real-world evidence data to really identify what are those risk factors. And we found some new novel biomarkers. And with that information, we basically changed our protocols for our clinical trial completely. We changed the protocol design, the inclusion and exclusion criteria, to really make sure we are going to basically enroll the patients that are at highest risk to get the infection. Then moving on to evidence generation. So as you all are probably aware, evidence generation is very important in clinical trials. Historically, one would always have one experimental arm where we enroll patients that actually get the drug, and one control arm where the patients get the placebo, and then we just compare the two. And this is definitely going to stay that way also in the future. But what we're trying here is to collect additional data that kind of, we can present to regulators on top of this more standard data collection. So one quite novel idea is to have single-arm studies, which are studies where you basically just have one arm. It's just the experimental arm, so all the patients that you enroll in the study will go on the experimental arm, they will get the drug. And then what you do, you try to match these patients to patients in the real world, and you compare the two. And the goal of this trial is to evaluate if your treatment is better than the standard of care. We have done that recently in one of our multiple myeloma trials, and also worked really closely with regulators to bring this single-arm studies forward, basically. As I said, it's still very new, and we definitely have to also have better real-world data to really make that more available. Another point for evidence generation is we try to track or allow patients to track their diseases more at home. So one example is nocturnal scratch. It's for skin diseases, where patients actually scratch their skin at night. So we developed this sensor that can detect nocturnal scratch based on radio frequencies. So we can automatically collect the data. The data is sent to the physicians, or if we do clinical trials, it's sent to us, and the patient doesn't have to come into the hospital all the time. Another example, tests for Alzheimer's disease. So we also developed a test to diagnose Alzheimer's earlier. This test is based on 3 steps. One is it measures your gait, it measures your cognitive abilities, and it also measures, well, it's like a virtual reality where you are told to place some items, and then later you have to go in and retrieve those items to test your memory. And with this simple test, which takes 10 minutes at home, we are able to really identify preclinical Alzheimer's symptoms. So we basically are able to diagnose Alzheimer's much earlier. What is the advantage of these digital endpoints? Basically, we get more data from the patients because it's all standardized. There's a sensor, there's a test that they do regularly. The patients don't have to come to the hospital regularly anymore because we can collect the data at home. And that is particularly interesting for us as a pharmaceutical company because we want to move more towards decentralized trials where patients can stay at home and still participate in the trial. So then you don't have the patient burden, them going to the hospital in regular intervals. You also have the advantage that you enroll more diverse patient populations than if you really have on-site trials. And that brings me to the last section, trial operations. So currently, it's very difficult to enroll enough patients for a clinical trial, especially if it's like a rare disease or low prevalence trial. Normally, just 4 to 5% of patients are actually enrolled in clinical trials. Why is that? Because some of them don't hear about the clinical trials, and sometimes we just simply look at the wrong places. So to start looking at the wrong places, we now make use of real-world evidence data, meaning we actually check where are our patients. We can't identify individuals, but what we can is look at certain hospitals and identify this hospital has more patients of the patient population we are interested in than another one. So we're combining this real-world evidence data together with past performance data, and then build machine learning algorithms to predict at which sites would we enroll most of the patients of interest. This has been quite successful, actually. One of our immunology trials has been accelerated by 50% because of these models. Also, we have diversity, equity, and inclusion models, which allow us to predict where's the highest representation of diverse patient population at a given site. And here again, in one of our immunology trials, we had the highest rate of Afro-American population, which of course we are very proud of. And I also want to mention that this is not a pilot for immunology now. We are scaling this up to more than 50 trials in our portfolio, and it's really becoming a standard of how Janssen performs clinical trials. That brings me to my last slide. Here, I just wanted to kind of summarize. We at Janssen totally believe that data science will change healthcare completely. And for our drug development cycle, we think it will have a special impact in two areas. One is it will, it can either bring what was I going to say? It can increase the success that the drug comes to market. So as you know, there's a lot of drugs who may be tested along the way, but they just don't make it because they're not efficient enough or they are not safe. So data science can help with that to increase the probability that these drugs will actually go through the whole process, or it simply makes the process faster. So all our data science projects are kind of focused in this area to make better medicines for our patients. And while we have a lot of different projects, we kind of just scale what works. We really work closely with external companies and regulators to really deeply engage with the ecosystem, because we think that data science is a team sport. And we also try and fully integrate and democratize data science within the company, meaning we are actually putting scientists, like the biologists, chemists, together with data scientists to solve our biggest problems together. And with that, I would like to thank you. If you have any questions, I'm happy to answer.